Hello, everyone, and welcome to today's webinar, which is on the study design considerations for Phase 2A proof of concept trials. My own name is Brian Fox. I'm a research statistician here at nQuery, and I'll be your host for today's webinar. So, just to quickly touch on the agenda for today, I'll begin with just a very brief overview of Phase 2 clinical trials, just to kind of ensure that the terminology I'll be using is on the same page for everyone. And then in section 2, I will go through Simon's two-stage design and some examples. I'll extend it into Simon's admissible design. And we'll take a look at how this rather kind of basic design has become so powerful and so widely used. And then we will extend this into some more complex Phase 2A designs, like Bryant and Day and many more. And I will show examples of these within the software. And then I will briefly finish up with a discussion and conclusion section where I'll touch on some of the points raised during the webinar and give some more topics maybe for people who are interested in learning a bit more. You're very welcome to ask any questions throughout the course of the webinar. And a recording of the webinar along with some of the examples will be available after the webinar concludes. Okay, so just to quickly touch on it, today's webinar is brought to you by nQuery, and indeed, nQuery is the software that we'll be using for our examples later in the webinar. But nQuery is widely regarded as just being a very cost-effective and easy to use trial design platform, which can be an essential tool in optimizing your clinical trial. It has designs across every phase of the clinical trial framework. So yeah, we've been around for quite a while, and we're always happy to help or take any questions. So do feel free to get in contact if you'd like. So just to quickly touch on how nQuery is structured, there's essentially 4 different tiers to the software. With base containing our frequentist designs, plus containing our Bayesian designs, pro our adaptive designs, and then expert containing our trial milestone prediction tool. So most of the examples that we'll be looking at today are contained in the pro tier, although 1 or 2 are from the base tier. But yeah, just to mention that before we move on. So now let's get into part 1, where we'll discuss Phase 2 trials. So I'm going to assume that most people here are quite familiar with the phases of clinical trials, but just to give a very quick refresher and ensure that the terminology that I'll be using is very clear to everyone, I'll briefly go through the 3 pre-approval phases of clinical trial. Also, just to mention that there is a phase 4, but it's just for post-marketing approval or post-marketing evaluation. So phase 1 is the first in human trial and will generally consist of around 10 to 50 subjects. And the objective of these trials is generally just to ensure that the proposed drug or treatment is safe for human consumption. So an example of something like this, phase 1 is usually trying to find the maximum tolerated dose, or MTD, which is basically equivalent to the highest dose, which can be administered without causing severe side effects. So this is often taken to be the maximum dose in the later phases of the clinical trials. Phase 2 then relates to the proof of concept and dose finding processes or phases, and would generally consist of somewhere between around 30 and 1,000 subjects. I'll discuss this in quite a bit more detail in just a moment, but just to mention that this is usually split into 2 processes of proof of concept and also dose finding. And these are usually treated as two distinct phases, where proof of concept is labeled as phase 2A, and then dose finding is labeled as phase 2B. And then finally, phase 3 relates to the classical kind of confirmatory trial, where one is trying to show that the drug or treatment is indeed effective over the existing standard of care or placebo or whatever it is. And then also show that it's safe, at scale. And this is obviously the phase at which regulatory approval becomes maybe most prevalent. Okay, so now, as mentioned, I'll just quickly give a bit more detail on phase 2 trials. So phase 2 studies have two main goals, as mentioned, proof of concept and dose finding. Historically, the proof of concept and dose finding objectives have often been evaluated in separate trials, with proof of concept established first, and then a dose finding study used to select doses of further interest. In this framework, the proof of concept trial was referred to as phase 2A, and the dose finding as phase 2B, with the proof of concept and dose finding studies using very different statistical methodologies. So, the goal of the proof of concept step is to establish that changes in dose do in fact lead to significant changes in the efficacy and or safety endpoint, and that at least one dose is of clinical interest. The emphasis here is on the kind of quick evaluation of the treatment and determining whether there is sufficient evidence to warrant further investigation of the treatment. Typical tests or designs that would be encountered at this phase would be one sample designs, so the Fleming test and Simon's two-stage design, which we'll be looking at later. Then the dose finding step, on the other hand, is concerned with selecting one or more kind of effective and safe doses for evaluation in confirmatory phase 3 trials. The emphasis in this step is on estimating the true dose response curve in order to ensure that the correct model and doses go on for further study in the phase 3 trial. Also, just to note that there is increasing emphasis on seamless designs, where the dose chosen from phase 2B trial and the information from that trial is automatically carried over into the phase 3 trial. So, something like MAPS designs. But yeah, some of the very common designs at this stage would be the Cochrane kind of Armitage test for proportions. MCP mod would be common at phase 2B. And then also, as mentioned, the multi-arm, multi-stage type designs. Okay. So now just to quickly touch on some of the trends that have kind of come about for phase 2A trials. As has been a kind of tumultuous couple of decades. I suppose the biggest shift over the last kind of decade or decade and a half has been the move away from purely frequentist two-stage designs towards more Bayesian and kind of continuously adaptive frameworks. So in terms of the Bayesian type designs, the kind of Bayesian predictive probability type designs have become quite popular. Something like the design of Li and Liu from their 2008 paper, I think. And, it is something that allows continuous monitoring rather than just looking at fixed interim looks. And I think that design also allows you to stop for futility or efficacy after every single patient, which is obviously, quite appealing, as you can imagine. And then another example would be something like the Bayesian optimal phase 2 designs. So BOP2, as it's often known, or often referred to as. And then also there's BOP2C, which is kind of an extension beyond that. And they have become increasingly common as well because they kind of combine the Bayesian flexibility with the sort of operational simplicity that a lot of clinicians would really enjoy or really want. um, then we also have sequential Bayesian type designs, kind of similar. They're also becoming a lot more popular. And then also Bayesian hierarchical models, actually, for something like basket trials, where these would kind of borrow data across different cohorts, allowing information to be shared across different subgroups, while also protecting your type 1 error, of course. Um, and then multi-stage designs, going beyond science design. I'll be showing a lot of these later in the webinar, so maybe won't talk about it too much here, but there's kind of a lot of relatively simple and easy to understand extensions to science design, which can combat a lot of the issues that are present at the phase 2A stage. But then I suppose the kind of one of the more kind of important structural changes that has come around in phase 2A, maybe oncology in particular, has been the development of master protocols. I suppose the kind of regulatory approval around that. So stuff like basket trials, umbrella trials, platform trials, they've all become quite a bit more popular. Primarily, or well, not primarily, but partially due to the fact that the FDA has become, or a lot of regulatory agencies, I should say, have become a lot more accepting of these types of designs. Something like the FDA's Project Optimus is probably worth mentioning, not specifically related to master protocols, but it's just worth mentioning because it's been pushing sponsors to kind of formally optimize dose selection within oncology before phase 3. So rather than relying on the maximum tolerated dose from phase 1, this is just kind of, I suppose, kind of creating a renewed interest in randomized phase 2A or B, I should say, dose ranging designs and kind of response adaptive randomization, which is obviously a big change from the kind of single arm and maximum tolerated dose based designs that people might be used to. Then we also have ICH E20, which is a guideline on adaptive designs, which is, I think, being finalized to the best of my knowledge very soon. And this will kind of provide a unified international regulatory framework for adaptive designs, not just phase 3, but also phase 2. And then finally, I suppose we have a decentralized and kind of hybrid type trials. This kind of is to do with allowing in, home visits or data from home visits, but, remote consent kind of things. And, telemedicine assessments. These are extremely powerful kind of tools if used correctly, and they're becoming a bit more popular as well. So then the final thing to mention, I suppose, is that we're kind of, or in this area, we are kind of moving beyond the traditional binary response endpoint. So classical designs like science design tended to assume a single binary endpoint, by which I mean, like a response being yes or no, or being progression free at a certain time point. And at the moment, most or a lot of designs are moving beyond that. So some examples of that would kind of be using time to event designs, or endpoints, I should say. So you're essentially using progression free survival or overall survival directly, rather than using a dichotomized kind of landmark rate. And this matters more for immunotherapies, since they produce, kind of long-tailed responses that might not necessarily fit a binary response framework. So then we also have stuff like a co-primary endpoint or a joint efficacy toxicity kind of endpoint, which is what Bryant and Day, which we'll be looking at later, have. But then There's also lots of other designs, like there's the Tall and Cook design, I believe. I think it's called FTOX. And that's, it's a Bayesian kind of analog to Bryant today, essentially, but it's very flexible. But then we also have composite endpoints and multi-outcome designs, which are becoming more popular. And these are a bit more relevant for kind of cell and gene therapies, where efficacy, durability, and toxicity all need kind of, I don't want to say equivalent, but, similar levels of consideration. Okay, so now let's have a quick look at some of the challenges in phase 2A. So, just in relation to the kind of, response, I suppose one of the main challenges that exist is that, there's often very delayed or atypical response patterns, especially from immunotherapies. And, stuff like pseudo progression or, delayed onset or a long tail in the response breaks the assumption that, an early landmark, let's say 8-week progression-free survival is going to be informative about the final outcome. So that kind of undermines the classical Simons type design. Another consideration would be that, the reproducibility and the translatability, if that's a word, or let's say the translation to phase 3 trial. A non-trivial fraction of positive phase 2A trials actually fail to replicate in phase 3. So that kind of, I suppose, it brings forward the debate about whether current designs are too permissive, and whether the field maybe needs stricter phase 2A go or no go criteria. Another consideration is complexity. So, obviously, if you're running lots of parallel kind of phase 2 cohorts, whether it's in a man's trial or whatever it might be, this does invite the issue of, multiplicity of testing and may inflate your error rates. Whereas the classical designs treat each cohort independently, and so they can get around that. But something like the man's group sequential design can easily get around that, so it's not a major issue, but it is a consideration. Another point to note, actually, on the topic of MAMs is that stuff like basket, umbrella, and platform trials, they do offer a lot of efficiency, potentially, but they do require very sophisticated infrastructure, stuff like cross-site coordination. You might need a data monitoring committee, or you will need almost certainly, to perform a bit of oversight. And, this is something that some sponsors or sites just aren't equipped for. Then just to focus in on it in particular, because it is a huge part of the phase 2A design landscape, is the area of oncology. So just to specifically focus on that, some of the main considerations or challenges that they face are as follows. So in terms of safety, it's obviously present in lots of different types of trial, but the issue is usually a bit more prevalent in oncology trials due to the likelihood of mortality being increased and the relative sensitivity of the patient to a new therapy. As a result, it kind of becomes more important to assess the safety of a proposed treatment in as timely and efficient a way as possible. This slightly ties into the next issue, which is often more prevalent with rarer forms of cancer, which is obstacles relating to patient recruitment. As you can imagine, patients may be quite reluctant to enroll into a program that bears too much uncertainty and maybe not enough data. That may be due to concerns around the effectiveness of the treatment at that stage, which is obviously a lot more prevalent at phase 2, considering that, there's no phase 3 trial ongoing that has the phase 2 trial data to kind of lean back on. So it's a bit more bit more of an issue at that stage, or at phase 2. But then I suppose the largest issue from a phase 2 oncology trial perspective is the relatively high failure rates, especially in comparison to other therapeutic areas. There was a rather comprehensive 10-year study, I think it was released in 2016. It's in the references anyway at the bottom. I think it was Bio et al. But it reported anyway that 5.1% of new cancer drugs that were tested in phase 1 eventually received FDA approval. So that was the lowest rate out of all the 14 different therapeutic areas that they studied. And this compared to rates of around 20% for areas like cardiovascular therapies. And in addition, I think the success rate, I hope I get this right. I think the success rate from phase 1 to phase 2 was around 63%. And then it dropped to around 25% when you went from phase 2 to phase 3. And that was compared to an average phase 2 to 3 progression rate of around 35% in the other therapeutic areas that were in that study anyway. Okay. I suppose the final issue I'll just quickly touch on is the idea of time constraints, since this is a lot more prevalent with oncology. Obviously, drug development isn't an overnight process, and it can take 10 years for some drugs to gain an approval. So that's potentially a lot of lives lost due to the delay in approving a drug, which is likely to be effective. And so as a result, oncology trials often have very short durations compared with other areas. And that bio study I mentioned found that oncology drugs had twice the rate of first cycle approval than something like psychiatric drugs, I think it was. And so oncology drugs were also approved the fastest of all 14 therapeutic areas that were given in that study. And, obviously this increases the time constraints and places a lot of additional pressure on the need to, optimize your study design in order to evaluate the drug in our trial, or treatment, I should say, in as efficient and timely a manner as possible. Okay, so all of these issues, and there's many, many more, this kind of just shows the need for improved trial design choice at this phase 2. Since these studies, they really need to be designed, performed, and reported in order to allow accurate interpretation of results and to obtain the best quality data to inform unbiased decisions at the phase 2 stage, or phase 3 stage, I should say. Okay, so that was a lot of background, but it'll go a lot faster from here. And now let's just quickly touch on Simon's design. So Simon's design is a 2-stage phase 2A design, which facilitates stopping early for futility at an interim look. So it was proposed in a paper by Richard Simon in 1989. And as mentioned previously, it has been a very common design choice ever since. So the design is based on the method of testing a null hypothesis of poor response proportion against an alternative hypothesis of a good proportion of responses. So a poor proportion here is defined as the largest proportion of successes, which, if true, would clearly indicate that the drug or treatment is clearly ineffective and does not warrant further investigation. Therefore, this can be considered as the proportion of successes below which a drug would be considered poor and be rejected. Similarly, the good response proportion, under which the alternative is assumed, can be defined as the minimum proportion of successes, which, if true, would indicate that the drug has a high level of efficacy and that does warrant further investigation. So this can be considered the proportion of successes above which a drug would be considered good and be recommended. A result of both the interim analysis and the specification of these proportions is that a specific cutoff value for the number of responses required in order to stop the trial at the interim look can be obtained, both for the interim analysis and obviously the final analysis. So this results in a set of 4 different parameters, combinations of which must be searched over in order to find the design which satisfies the specific design criteria desired. So in Simon's paper, and indeed also in nQuery, there are 2 main choices available for this design criteria. The first is the optimal design, which refers to the design which minimizes the expected sample size while also satisfying the error constraints. And then the second option is the minimax design, which targets the design which minimizes the maximum expected sample size. And these should, hopefully become a bit clearer when I take a look at the example within nQuery in just a minute. But just before that, I'll quickly bring your attention over to the graphic on the right over here, since it probably explains it a bit better than I'll be able to with words. But this just shows the individual steps or kind of processes that are involved in this Simon's type trial. So you recruit N1 patients, and then at the end of stage 1, you look at the amount of responses you have. If you have less than R1 or greater than equal to R1, then you either stop for futility or you continue and you recruit the second half, or sorry, not half, the second cohort of patients, N2. And then at the end of the study, so at the end of stage 2, you have your kind of final cutoff, which is denoted by OR here. And at that stage, you can decide whether to recommend or not recommend the drug or treatment for further evaluation. Okay, and so just to very quickly point out actually that one of the main benefits of Simon's type design over other phase 2A proportion endpoint designs is that it utilizes the exact binomial distribution for the response proportion rather than using a normal approximation, for example. And that just means that it's extremely accurate, although it is quite computationally intensive, as we will see later. Okay, so this example here is based on a trial that was testing the effect of this combination therapy here for treating patients with metastatic prostate cancer. And this trial in particular did employ a Simon's type design, a summary of which is shown on the left here. And note that this design will be extended out for our future examples, so I'll just spend a little bit of time here to try and give a bit of detail on what's going on. So the layout for the trial was as follows. They planned to have a first stage of the study, which saw the enrollment of 15 patients. And after the responses of these patients are recorded, and the total number of responses seen up to this interim stage are kind of added up, that is then compared to the cutoff value of 8, where if 8 or fewer responses are observed, then the trial will be stopped for futility. And if 9 or more are observed, then patient accrual will continue up to a total of 43 patients. So yeah, an additional 28 patients are kind of recruited. And then out of these 43 patients, at the end of the trial, if 26 or fewer responses are observed, then the study will find for futility and no further investigation of the drug will be recommended. And if 27 or more responses in total are observed, then the drug will be recommended for further investigation. So, in addition, they note that the expected sample size for the trial is around 23.5. And we'll see how that figure is calculated in just a moment. And interestingly enough, actually, this trial was stopped for futility. as no responses were found in the first 8 patients. So even if the remaining 7 patients in the first stage all had a response, then the total number of responses in that stage would still have been lower than the cutoff value of 8. And so the study was stopped. So yeah, now let's have a look at what this looks like in nQuery. So this is just what nQuery looks like. Hopefully some of you will be familiar with it. And we have lots of other webinars going through how to use nQuery if you want to take a look at any of those. But the design that we're interested in is this one here, Simon's design. And so up here, we have the main table, the top left. On the right here, we have our help cards, which will give you some information on each of the different rows. And then we'll see that our output will appear down below here in just a minute. So the first few parameters here are just the inputs required for this design. And we can, if we were to open up the example slide from earlier, we would see that the significance level was 0.05, or 5%. They're using the optimum design, which is the default here. They specified a maximum ineffective proportion of 0.5, and that is essentially the good response, or sorry, a poor response proportion. And then the good response proportion, which is labeled here as the minimum efficacy proportion, was 0.7. And now before I enter in the power of 80%, I'll just quickly touch on the fact that this design works in a slightly peculiar way. So you can see here we have our optimum search value, and that's due to the fact that, as mentioned, Simon's design in the optimum case works on binomial enumeration. So we're getting the exact binomial result from the distribution for the probability of early termination. And given that the optimum design is the design with the lowest maximum expected, the lowest expected sample size, which still satisfies the error constraints. Once you have your probability of early termination, you can find out your expected sample size. And so the way the algorithm works is by searching upwards from its current lowest expected sample size design by this number until it doesn't find a new lower expected sample size. So I know that's a little bit complicated or a little bit convoluted. And so what I mean by that is that once you have your combination of the total sample size, capital N, your final cutoff value, which is R, your first stage sample size, N1, and your first stage cutoff, R1, so you have those 4 parameters, along with your significance level and your power and your proportions, you can calculate the expected sample size. And in fact, actually, so you don't even need the power, but once you can verify that that particular combination of those 4 parameters satisfies the error constraints, you can take the expected sample size and then search upwards in such a fashion, until you find the optimum design. So in that respect, it's kind of like finding a local minimum, which is why this design is a little bit kind of difficult to use, or can be a little bit cumbersome. You'll see that it gets more and more difficult as we progress through the other examples. But yeah, it's just worth mentioning that, since it's quite important to know how these algorithms actually work. Okay, but now if I go back and I enter in our power, we can see that the study runs straight away. And you can also see that these parameters here, 43, 26, 15, and 8, all map on to our earlier values. Just to quickly go back. So 43, 26, 8, and then 15, they'll map on here. And then our expected sample size of 23.5, you can see we also have that here. So yeah, essentially to get this, all you're doing is multiplying your early termination by the first stage sample size, and then, multiplying the probability of not. you know, Essentially it's quite simple. It's this formula here, which is probably going to describe it better than I can with words. But it's very simple. Okay. So now let's go back and actually have a look at Simon's admissible type designs. So this is actually from a Mander 8L paper from 2012. I don't know if labeling it Simon's admissible designs is a bit unfair, but it's a relatively simple extension of Simon's design. So as we discussed, the minimax and the optimum are two kind of extremes. So the optimum minimizes the expected sample size, the expected sample size under the null, which allows for a higher maximum sample size, whereas the minimax minimizes the maximum sample size, which can result in higher expected sample size compared to the optimum. So this is where the admissible design or admissible designs can come in. So these are kind of compromises between these two. And so it's a kind of range of designs that both satisfy the error constraints and are within the minimax and optimal designs in terms of the maximum sample size, I should say. So the trade-off is kind of, you can have a slightly larger expected sample size, but have a slightly smaller maximum sample size, or vice versa. And that can kind of be shown down below here. But just to quickly show how this would come about, we can go back into nQuery, and you can see that it's very simple. This is a extremely quick calculation, 0.5 and 0.7, I believe it was. You can see there that we get back our optimum design here. So 8, 15, 26, and 43. This maps onto the earlier example, same expected sample size, but now we can see both the minimax and an admissible design. So yeah, in this particular example only has one admissible design, but there could be any number. Usually it's between 3 and 4, roughly. But yeah, it's quite a useful tool. It gives trialists a lot more flexibility about the type of design to choose, and is relatively appealing, I can imagine. Now, before we move on, just another type of Simon's design, since essentially his name is on it. That's worth mentioning, is just the pick the winner design. I believe this is from a paper in 1985, so quite a long time ago. But it's very useful. It can calculate the probability of selecting the best treatment arm or group in a phase 2 trial that has multiple arms. So yeah, feel free to take a look if you want. It's quite an interesting design. And if someone references Simon's design, usually it's not this. Usually this would be referred to as Simon's selection design. Okay, so moving on, let's have a look at some more complex phase 2A trial designs. So the first one we're going to take a look at is the Bryant and Day design. This design is based on the 1995 paper, which essentially expands upon Simon's design by incorporating a co-primary endpoint for both efficacy and toxicity. So this design is very similar to Simon's design, except that at each analysis, there's going to be an additional option to stop the trial if there is unacceptably large levels of toxicity. And I know that this is generally done on a sort of ad hoc basis, but it is useful to have this toxicity assessment incorporated directly into the study design itself. In addition, this design aims to kind of tackle the uncertainty in the level of toxicity being assumed for the treatment at the outset of the trial. As obviously at phase 2, only the relatively small toxicity data from phase 1 is likely to be available. So on the right here, we see the layout of this design, which is shown from the original paper by Bryant and Day. So this looks a little bit more complex than it actually is. Basically, there are 6 possible outcomes from the trial. So at the end of the first stage, the trial may be stopped for futility, for toxicity, or for both. And then if acceptable levels of toxicity and efficacy are encountered in the first stage, then the trial will proceed to the second stage, at the end of which the trial may find for futility, toxicity, or both, or else the levels of both may be found to be acceptable, and the trial recommended for further phase 3 study. And obviously, also here, the specific toxicity cutoff values are specified similarly to how the response cutoff values were required for the Simon's design. So that will hopefully make a bit more sense when we take a look at the example in just a moment. But just to mention before that, however, that similar to Simon's design, the design criteria available are the optimum and minimax choices. So now let's delve into an example. So as mentioned, we'll just be expanding upon our previous Simon's design example to hypothetically see what the researchers might have done should they have chosen to use a Bryant and Day design for their trial. So on the left here, we see a summary of the parameters required for the Bryant and Day design itself. And many of these are just taken from the original example, like the acceptable and unacceptable response rates and the power. However, some additional parameters are required, like the acceptable and unacceptable levels of non-toxicity, and also an additional type 1 error rate constraint for the toxicity element of the design. But yeah, once again, this will hopefully make a bit more sense once we look at this in nQuery. So here we have a Bryant and Day design. You can see that it's pretty similar to Simon's design, or looks pretty similar at least. But the main difference is just the toxicity significance level and the acceptable and unacceptable non-toxicity rates. And these can basically just be considered as the minimum non-toxicity rate, which, if true, would clearly indicate that the drug or treatment has an unacceptable level of toxicity. And then the unacceptable rate as the largest non-toxicity rate, which, if true, would clearly indicate that the drug or treatment has an unacceptably large level of toxicity. I know there's a lot of double negatives going on, so it's getting a bit confusing, but hopefully that makes sense. Now, the toxicity significance level is a little bit more complex. It's kind of explained properly here. So this is the probability of erroneously recommending a treatment that is unacceptably toxic. And then we can see that the hypothesis test under consideration here is just kind of shown, and this kind of shows the co-primary endpoints analysis through this simultaneous hypothesis structure. But anyway, let's go back and we'll enter in our parameter values from the slide before. Hopefully I can remember them. 0.5, I think. We'll know soon if I haven't. 0.95, and then once I enter in the power, this should start going. Now, due to the additional complexity with this type of design, it will take a fair bit longer to run. So I have one already finished here. Yes, that has the same values. That's good. I didn't make a mistake. So you can see that there is a slight increase in the sample size. So this co-primary endpoint has resulted in a bit of a kind of power loss or sample size gain, whichever way you want to look at it. And it is, this is very similar to Simon's design in terms of there's just cutoffs. It's the first stage cutoffs, final cutoffs, and similar for the sample size. But then you can see that the probability of early termination has actually increased relative to the earlier Simon's design example, just marginally, actually very marginally, yeah. But yeah, you can see then the exact error levels as well, which were also available in Simon's design. But yeah, this is relatively self-explanatory, although it is relatively complex in the background and maybe a little bit difficult to get your head around the hypothesis testing framework of it. But just given time, I'm going to move on to another complex phase 2A design, which is the Lin and Shih design. So this design is, a bit of a more complex expansion of Simon's design. It aims to tackle the uncertainty encountered in the specification of the response proportion that is to be used under the alternative hypothesis at the outset of the trial by allowing the user to specify two choices for that proportion. The first being an optimistic estimate and the other being a more pessimistic estimate. So the information that is produced and examined at the interim stage of the trial is then used to choose between powering the overall trial under either the optimistic or the pessimistic choice. Where obviously, if the number of responses at the interim look is below a cutoff, a certain cutoff, then the trial will be stopped for futility, similar to Simon's design. Except that if that number of responses is between that cutoff and another higher value, then the study is powered under the pessimistic estimate for the response. And then if it is above this higher cutoff value, the more optimistic estimate is used. So, due to the additional complexity with this type of design, there are multiple optimal design criteria available to choose from. Although I will explain these in a bit more detail in the example to come, and that hopefully should make it a bit clearer. And just to note that this formula on the right represents just the probability of rejecting the treatment, moderately similar to Simon's design earlier, but the calculations involved are quite a bit more complex. So yeah, this should look familiar at this stage on the right here at least. This is once again extending that example, but most of the parameters are very similar to Simon's design example from earlier, but we will be assuming that instead of the researchers saying the minimum efficacy proportion, or the good proportion, as we called it earlier, instead of assuming that that's 0.7, we'll be saying that they think a value of 0.7 was a pessimistic estimate for it, and that 0.8 would be a more optimistic estimate for it. So in order to make the discussion of this design a bit easier, the case where the pessimistic choice is found at the end of the first stage would be known as case 1. And similarly, the case where the more optimistic estimate is found to be justified will be known as case 2. Also note here actually that the Lin and Shih design has the flexibility to specify different type 2 error rates for each of these two cases, which may be an advantage, rather than strictly being held to the 80% power rate. It's kind of up to you. But yeah, now let's have a look at this within nQuery. Okay, so this is essentially what the Lin and Shih design looks like. It's relatively complex, or looks relatively complex. There's a lot of rows here. But at the heart of it, it is very similar to Simon's design. But yeah, so rather than filling out all of the parameters, since that will take quite a while, I have this one already filled out. So you can see that similar to before, we have our maximum ineffective proportion, but then we have our two different cases, case 1 and case 2, being our pessimistic and optimistic cases respectively. We're going to assume the same type 2 error rate of 0.2. I find it hard to think of a situation where you'd have different ones, but it's possible anyway. The maths allows it, which is nice. But yeah, so if you were to proceed through, and we're going to use the type 1 design criterion here, which is essentially like an optimal design, let's say. So it corresponds to the feasible design, which is to say the design which satisfies both the type 1 and type 2 error constraints, which has the lowest expected sample size under the null hypothesis. So the pi 0 in the maximum ineffective proportion. So that's quite a tongue twister. It's a little bit difficult to get the head around, but essentially there are 4 different optimality types or criteria. Type 2 being the feasible design in which the maximum expected sample size over all cases is minimized. And so it's kind of like the minimax essentially is what I would think of it as, but it's a little bit different. And then type 3 being the feasible design in which the maximum of m and n, being the case 1 and case 2 maximum sample sizes respectively, are minimized. And then the expected sample size under the null is minimized within that range. Similarly, type 4, it's the exact same, except that it's minimized. The expected sample size is minimized under every scenario. So both alternative hypothesis and the null hypothesis. So yeah, a little bit complicated, but I believe type 1 as well was recommended in the Lin Shih paper. And you can see that using this, we get the exact same sample size as our Simon's design. So all these match 43, 26, 15, and 8. 46, 26, 15, and 8. Due to the fact that this is kind of like running 2 simultaneous Simon's designs. In reality, it's a bit more complex than that, but that's maybe an easy way of thinking about it. So yeah, let's keep going. So now let's have a look at Litwin's design. So Litwin's design, or I should say Litwin, Wong, and Hugh's design, is from a 2007 paper. And it essentially just extends Simon's design to the progression-free survival endpoint. Although to say it just does that is a bit of a trivialization. It does a lot. It goes well beyond Simon's design in many respects. So the design itself is as follows. So similar to Simon's design, there's a first stage sample size of N1, and that amount of patients are recruited initially and then followed until each has been evaluated at the early time point. Let's call it T1. And if a sufficient number of these patients are progression-free at this time, then the trial continues and a second cohort is recruited, taking the total number of patients up to the maximum sample size, where these are evaluated at the final time point, T2, and otherwise the trial is stopped for futility, I should say, at the first stage or the first time point. And also, it's worth mentioning that the patients from the initial cohort who are progression-free at that time, at T1, I should say, for the first time analysis, they'll remain on the study and they're reevaluated at the final time point, T2. So, yeah, it's actually relatively simple. It doesn't sound it to say it out loud, but it is, I promise. So another benefit of the Litwin design is that it extends Simon's design to both the null and alternative hypothesis. So an optimum design is available under both these hypotheses, whereas in the Simon's design paper, the optimum design is only available under the alternative, I believe. So, in fact, there are actually 4 different design criteria or criterion available. So there's the optimum design under H0, or the null hypothesis, optimum design under the alternative hypothesis. There's the usual minimax type design, but then there's also this custom type design, which I'll explain within nQuery. Actually, I suppose it's pretty easy. Easiest to go into that now. Probably make it a bit easier. So, yeah, just to explain, I'll go back and explain this example in just a moment, but just to explain. So within nQuery, this is the design criterion. You can see that we have a short explanation of each of these. And the custom design since it's what we're going to be using to replicate the results from the Litwin paper. This is essentially the smallest maximum sample size, where the first stage sample size is within 10% of whatever user specified proportion. So that's this parameter here. 0.5 is what we specified. But that proportion of the total sample size. So in this case, the total sample size is 28. So it will be within 10% of 14. So yeah, hopefully that helped. But yeah, this example, we're just going to replicate the very first example from the vast tables given in that paper. But we're going to replicate table 8, just because it has a nice alpha and power of 0.5, 0.8. And we're going to be assuming a lot of different probabilities here, but I'll explain those within nQuery. Just you'll have to take my word for it that these are the results we're aiming for. So this is obviously very different to Simon's design. Instead of having our kind of minimum, maximum, effective and ineffective proportions, we have our probability of progression-free survival up to the first time point under the null. So P01, similarly under H1, or the alternative. And so these are said to be 15% and 35% respectively. But then we also have the overall probability of progression-free survival up to the final time point of T2 under both the null and alternative. And this, using these 4 values, you're able to calculate the conditional probability of progression-free survival up to T2 under both the null and alternative. And then finally, you need to specify the minimum probability of early termination under the null and the maximum probability of early termination under the alternative. And so a little bit complex, but yeah. Once you get your head around it, it's actually pretty simple. So once those are specified, you can enter your power, and this can take a little while to run, which is why I've calculated all these already. But you can see that we get our sample size cutoff, first stage sample size and first stage cutoff of 28, 3, 15, and 2, which if we go back, we have 28, 3, 15, and 2. So just to verify that this is working. But then if you wanted to find the optimum design under the null or alternative, you can see that you get relatively similar results. Just a difference in total sample size of 2 here, and relatively simple to the custom design. And then also, if you go for the minimax design, you can see that it actually is the exact same in this particular case as the optimum design under the alternative. So yeah, just a interesting result in this case. But yeah, I won't spend any more time on that now, as I'm realizing I'm getting pretty close to running out of time. But just before we move out of nQuery, just to quickly mention that another type of design that you might be interested in is the Fleming group sequential test of one proportion. This is quite commonly used in phase 2A. You can see it looks relatively similar to Simon's design, where here we have 4 different looks, and that each of these looks you're actually allowed to stop for efficacy or futility. So we have our futility point here, so where we can accept the null hypothesis, or we have our rejection point where we can reject the null. So yeah, very, very simple. We also have our 3-stage design, which kind of goes beyond Simon's design and extends it to 2 interim stages. So it's just additional flexibility. And then we also have our FCV mod, for example, and then we also have our MANS group sequential trials. So if you're looking at phase 2B, just to mention that they're in there, they're very interesting. They're really cool maths going on in the background, or statistics. So yeah, just to mention that they're in there as well. So yeah, just to very quickly wrap up, phase 2A trials have a very important role in terms of screening out ineffective treatments prior to our very costly phase 3 trials. There are lots of challenges facing phase 2A trials, such as delayed response, issues with error control, there being high failure rates, etc., etc. And adaptive and Bayesian designs can be used to combat a lot of these issues and are being used to combat a lot of these issues. But then also some kind of more simple multi-stage designs can offer very powerful and useful solutions towards some of these issues. Okay, so if you'd like to take a demo or have a look at anything else that I've shown here today, feel free to go to statsolves.com/nQuery demo, and you can get a free trial. Also, do feel free to get in contact with us here at StatSolves. And once again, thank you very much for listening. Bye.